Okay, everyone good with seeing my screen and hearing me? Awesome. I hadn't, oh, that was from earlier. Okay, cool. All right, let's keep going on. Oh, we were on this one. So we had just finished up talking about these first three types of normalization methods. This is just different types of calculations. You guys can play around with these calculations based on the total number of reads that we have in our gene counts. And then if you have any questions about how these are calculated, if you play with them, you can go ahead and let me know. But I'm not going to spend too much time on these types of normalizations just because it's not the ones that we use at GeneLab. The normalization method that we do use at GeneLab is called the median of ratios method. And so this we use through the DC2 tool, which is an R-based tool. And what this does is it calculates a size factor for each sample by dividing the median ratio of all gene counts. So using the median, it takes away the possibility of skewing the data based on like a contaminant, right? That'll have a crazy number of gene counts, like what we saw when we looked at our duplication level in that FASTQ report. And so it'll calculate the size factor for each sample by dividing the median ratio of all gene counts by the geometric mean of each gene across all samples. The raw counts for each sample is then going to be divided by the sample-specific size factor for each gene. So a size factor is created for each gene based on the median ratios of the expression across all samples. And Lauren is going to get into that in quite a bit of detail in her stats lecture so that you guys can understand the math behind that. And by doing that, you can now account for both sequencing depth as well as RNA composition. And so that's why this is a really good tool to use to be able to compare gene counts between samples. However, if you're not accounting for gene length, you of course probably wouldn't want to use this to look for differences in expression of genes within one sample. One thing to note about that though, the tax import tool that I had told you guys about that we use in our usual pipeline at GeneLab, to pull in, so we pull in directly into R, the genes.results files using tax import. And when you do that, that actually normalizes based on gene length using the effective gene length from the RSEM genes.results file. So using that function through pulling in the data through tax import, you actually do normalize your data based on gene length, and then you can do those comparisons in any way you want. And that's actually what we do in the pipeline in GeneLab. So the data that we have, you can make those types of comparisons. And then just one more tool here that I want to talk about is the trimmed mean of M values, or TMM. So this is used by a tool called EdgeR. And this one works somewhat similarly to median of ratios. So it also has that caveat of not accounting for gene length. But there's been this new way to run TMM where you do account for gene length, and that's called gene length corrected TMM. And so in this way of normalization, the total read count, or the total reads per kilobase. So basically, if you do the GETMM version, instead of starting with total reads, you first normalize those reads based on gene length, and then you correct it by a normalization factor. And then that normalization factor in TMM normalization is calculated using a weighted mean of the log expression ratios between samples after trimming. So taking the log also kind of accounts for that large difference that could happen with the contaminants. And then that's scaled to per million reads. And so that will account for sequencing depth and RNA comparison if you do just TMM regularly, or if you do the GETMM and take into account that gene length by normalizing for the gene length before you start your TMM. Then you can account again for all three of those issues that we talked about and be able to use it for within sample comparison and between sample comparison, and within group comparison as well. Okay, so Lauren's going to dive in a lot more in that median to ratios method. So that's really as much as I wanted to talk to you guys about this stuff. So if you guys want to read about it and read about these tools, and then if you have any questions for me, you can bring them to me tomorrow. But are there any immediate questions right now? I have like 3 more slides left. Okay. So yeah, if you guys take the time to look at those and have any questions, by all means, send them my way tomorrow. So the type of between sample normalization used greatly impacts differential expression analysis. So I know I mentioned this earlier, but just want to harp on it again, that the type of tool you use to normalize is really going to have the most impact on your data. And so as I mentioned, we're going to discuss the DC2 normalization method in a lot greater detail. Lauren's going to talk to you guys about that in a stats lecture that she put together. Now, after we account for these differences in our count data, and then we have normalized count data, we can then begin the last step of the pipeline, which is differential gene expression. And so as we're keeping track of our pipeline, so now we've gone all the way through all of these steps, normalized our counts, and we'll now be ready for differential gene expression. And again, we use DC2 for this as well. And so this is common. So like with Limavoom too, that's another tool where it does both the normalization and the differential expression in one. And so DC2 is a tool that you do both normalize and you can do differential expression with those normalized data. And so now we'd be ready to determine the probability of each gene's expression being significantly different in one of our groups of interest through a pairwise comparison. So comparing our flight samples to our ground control samples in our data set, or we could do a multiple comparisons testing and kind of look at general differences across all samples. And so there are, again, a bunch of different tools available for differential gene expression analysis, and they're going to differ in the type of distribution that they use to create a model to fit the count data for statistical testing, and also the statistical test that they use to determine if there are significant differences between your groups. And again, this part of the analysis requires quite a bit of statistics, so Lauren will go through this stuff with you. I just wanted to show you quickly the few different tools that can be used to do differential gene expression. So again, we're going to be using DESeq2, and that uses a negative binomial distribution model. And the statistical test that it uses, it'll use a wald test for pairwise comparison, which is what we're going to be doing with our data. And we can also use a likelihood ratio test for multiple comparisons testing. We do that in the GeneLab pipeline, but we're not going to do it in our bootcamp. This time around, because we mostly focus on those pairwise comparisons anyways, so we figured those were more relevant. You can also use Limavoom to do differential expression, so this uses a different assumption model, so it's similar to a t-distribution with an empirical Bayes approach, and they use a moderate t-test to do their statistical testing. There's also EdgeR and BaseSeq, which both use negative binomial. However, they use different statistical tests. EdgeR uses exact test, and BaseSeq2 will use posterior probability through the Bayesian approach. And EBSeq uses negative binomial beta empirical Bayes model, so kind of both of these models. And that statistical test they use is the same as the one for BaseSeq, so posterior probability through Bayesian approach. I gave you guys links to all of these tools, and I'm going to give you another reminder that if you have not finished your formal education, I cannot recommend enough taking statistics courses. There's a lot of statistics in bioinformatics. A lot of what we decide of what's differentially expressed or what changes are significant in our data is based on statistical models. So the more you understand and know statistics, the better bioinformatician you're going to be. So with that, I'll leave you with the fun stuff here. So once we do our differential expression analysis, a great way to take a look at what genes are differentially expressed between groups is by generating pretty pictures. So data visualization is going to be used to help us interpret count data and differential gene expression data. And so we're going to be generating these 3 plots in particular during the bootcamp. So we'll be generating a principal component analysis plot, a clustered heat map, and also a volcano plot. So I'm not going to get into these at all because Lauren is going to cover all of them in her lecture and also in her Jupyter notebook. And with that, does anyone have any questions about the RNA-seq overview? This was a beast of a lecture. It covered everything from the theories and technologies that led to the awesome revolution in sequencing by synthesis brought to us by Illumina, and followed all the way through to data processing and analysis. So this was a ton of information, guys, and I'm super impressed with how well you guys have been keeping up with the material, and that's really obvious through all the thoughtful questions you guys have asked throughout this lecture and also this first notebook. So any final questions about the RNA-seq pipeline? Yeah, amazing stuff. Going to be rewatching these lectures multiple times. Cool. Yeah, it takes a while, guys, for this stuff to kind of sink in. But I think you guys are all on track to really know this stuff and know this stuff well, just from how much you guys seem to have absorbed just in this first pass. Okay, great. So I am finally going to give you guys a break from hearing my voice. One more question. From a biology standpoint, wouldn't differential isoform analysis be more useful than differential gene analysis? Good question, Felix. It completely depends on the question you're asking. So it depends on what you want to know. If you really want to, so if you have a gene that you have in mind and you really want to know about the different isoforms, right? So if your different expression of different isoforms has a different physiological effect ultimately on your samples, right? So let's say you know that if you have an increase in isoform A compared with isoform B, then you have an increased likelihood, let's say, of getting cancer. Then that's going to be something that you're going to want to take a look at. But if there really is relatively the same expression of all isoforms for a gene in the experimental study that you're looking at, right? So if you're looking at differences in spaceflight and you take a look at your isoforms for the gene that you're interested in and you see there's no difference in the level of isoforms relative level of isoforms being expressed between your flight and your ground control group, then that isoform resolution really isn't going to matter. You just want to see if the overall gene expression is going to be up. So good question, and it completely depends on the context of your experiment, on the nature of the genes that you're interested in, and on specifically the question you're asking. So sometimes it's going to be more useful to look at isoforms, sometimes it's going to be more useful to look at genes. Good questions. Okay, any other questions? Okay, I personally am very sick of hearing my own voice, so I'm sure you guys could use a break from hearing my voice too. So I am super happy to hand over control of the screen to Lauren, and she's going to take you guys through starting with the Jupyter notebook and then talk to you guys about the statistics that we use through her lectures. So go ahead, Lauren.
All right. Thank you, Amanda. That was an amazing couple of days. We all learned a lot, including me, and I've been doing this for a while. Okay, can everybody see my screen?
Yep.
Great. Okay, great. So the first thing I wanted to do before we move over to the differential gene expression analysis Jupyter notebook is thank you so much to Kevin for figuring out some code so that we can read in our RSIM genes results file in pandas. I just wanted to share another method, an additional method for doing so. When you look at the genes.results file, you can kind of see visually that it's a tab separated file or a TSV file, even though the extension isn't TSV, right? It's .genes.results. So you can actually read in this file using Pandas read CSV directly, and you can pass in the variable that we defined at the beginning for the path, RSIM output, because when we defined this variable, we didn't do it in Unix. This was just in Python. So this is just a Python cell, and we've defined all these paths. So these are all Python variables which we could also access in Unix because we define them in the kernel. So Python syntax is just let's put the path here outside, we use a plus sign, and then we give the file name as a string. And then because this is a tab separated file, we tell read CSV that actually it's not a comma separated file, it's a tab separated file using the sep argument. So, and then we can do .head, which will give us our nice little table there. So I just wanted to share an additional way that you can look at these files using pandas. All right, can you share the chat? Yes, of course.
And so just to point out, but before you move on to the next Jupyter Notebook, if you just want to remind everyone how they can export this one if they want to.
Yeah, for sure. So in order to export this Jupyter Notebook, you can go to File and Export Notebook as HTML, which will save all of the outputs. Or you can go to your file directory here on the left and just download it, which will download it as a Jupyter Notebook file in case you want to run it in Google Colab. And I know that I owe some people some documentation on how to get your R kernel up and running in Jupyter Notebook, and I will do that as soon as possible. All right, so let's switch over to the didn't appear in chat. Oh, that's because I sent it privately to Amanda. Thank you for pointing that out, Felix. There you go. All right, let's switch over to the next Jupyter Notebook, RNAseq DG JN0621. So open that up. So we're going to go through the first step of this notebook, and then I'm going to lecture a little bit on statistics, and then we'll come back to the notebook and do a little bit more statistics lecture. So we'll see how far we get today, but we have plenty of time to finish up tomorrow. All right, is the font big enough for everyone? Maybe I can make it bigger here.
Yeah, maybe make it a little bit bigger.
How's that?
That's great.
Okay. Okay. Great. So Amanda has already shown this workflow. We're starting with raw counts and we're sending them and sample metadata, and then we're sending them all the way through DC2 to perform differential gene expression analysis. Along the way, we're going to make some PCA plots, and I will talk about PCA plots more in the statistics lecture. And we're going to add in some more gene annotations beyond just the ensemble genes. And finally, we're going to do some fun visualizations and analysis. So this notebook is, unlike the last one, is written in R, so it's running an R kernel. And we, again, have a table of contents. So let's just jump right into the first part here where we need to load in our libraries. So remember from the R intro notebook that we need to import libraries if we want to use code or data packages that don't come with the base R installation. So thanks to Stephen Boring, all of these packages have already been installed on the Jupyter Hub, and we just need to import them. So let's go ahead and start running these cells. So first we have the tax import library, which Amanda pointed out we're not actually using in this version of the differential expression analysis, but it's a typical library that we use at GeneLab for reading in the RSEM genes.results files and normalizing everything by gene length. We then need to import the DESeq2 library along with a bunch of other auxiliary libraries that help out with the differential expression analysis. So let's run that. We get some output here, just letting us know some of these packages have a little bit more verbose output when we read them in than others. I always scroll through just to make sure that we haven't gotten any errors, we will notice that a lot of this text has to do with these packages are figuring out what other packages are the dependencies and whether they're already installed or whether they need to be loaded together with these packages. And then certain packages are masked, meaning that they've already been installed at a different version. And we have some specific versions here. We have some conflicts, which are not going to be a problem in this case, but can be a problem in other cases. All right, then we need to import a bunch of libraries that are going to allow us to perform data visualization. The first one that we're importing is called ggplot2, which is a very famous library for creating publication level graphics in R. And then ggfortify and ggdendro are sort of offshoots of ggplot2. We're going to use the complex heat map library to make our heat map. A tidy heat map is going to help us write it out to a file, and then the enhanced volcano package is really nice for making volcano plots. Amanda pointed out that it's really nice when tools just give you upfront what's the version, and complex heat map is one of the only R packages that actually does that. When you read it in, get the version, the GitHub page, lots of documentation and references, and what we're supposed to cite when we use it. And then finally, we're going to import two libraries, String and Panther, which are both databases with gene and pathway annotations that we're going to link up to the ensemble genes later on. All right. Did anyone get anything different from what I got here? Any weird errors? Any of these cells not able to run? Okay, excellent. So having imported, my cells don't seem to run. Okay, Veronica and everyone else, does it say R idle here? Down at the left-hand corner? Okay, so Veronica and anyone else where it says no kernel, click on it and just select the R kernel. Then you should be able to run these cells, but let me know if you can't. That was my mistake. I should have checked that everyone had their R kernel working. Okay, perfect. So the first step here is that we need to import the raw counts table and the metadata table so that we can tell DESeq2 what is the gene expression, and then what are the sample names and which samples belong to which groups, because we want to tell DESeq2, we want to compare the flight versus the ground control groups. So the first thing we need to do is, again, set up our directory path variables so that we have shortened variables to refer to where all of our input data is and where all of our output data needs to go. So let's go ahead and run that cell. And now that we have our paths set up, we can go ahead and import our data. So first we're going to import a metadata table. And this is a file that Amanda set up before the bootcamp that we're going to use to tell DESeq2 which samples belong in each of the groups, flight or ground control. And then secondly, we're going to import the unnormalized or raw gene expression data file, which we just created at the end of the FASTQ counts notebook. So if you recall, at the end of the FASTQ counts notebook, we wrote out the counts table to a CSV file, and we're just going to read that right back into our notebook so that we can pass it into DESeq2. All right, but first, let's import our metadata table. So this is new. We haven't looked at this before. We're using the read.csv function that we learned about in the R intro notebook, and we're assigning the output of this function to a variable called sample table. We can use the next cell to take a look at this metadata table. You can see it has one, the first column are the row names, which contain the sample IDs. These match and are in the same order as the counts table that we wrote out. It's very important that the metadata table have the samples in the same order as the expression table. And then the second column indicates the group to which each sample belongs. So obviously all of the flight samples and then all of the ground control samples. Now this is nice for us to look at, but we need to pull specific information out of this file and format it in a specific way so that R and DC2 can use this information. DC2 requires the metadata to be in a specific format. So let's go ahead and run the next cell to do this formatting. We can ignore the warning message here. And then using the formatted data that we generated in the last cell, we're going to define all the different group comparisons that we're going to use for differential gene expression analysis. So Amanda just mentioned for differential expression analysis, we can either compare two groups, for example, flight and ground control, or we can do a multi-group comparison. In this case, we're just going to be doing two groups. So DC2 specifically requires a set of what they call contrasts, which just means the groups that we're going to compare for differential gene expression analysis. So the next cell is some code which grabs the flight and ground control status of each, which grabs the different groups from the metadata table. In this case, it's just two groups, and sets up all possible contrasts. So if right now we have only two, so it's just going to be pairs of contrasts. If we had more than two, we would have multiple pairs of contrasts. So let's go ahead and set up these contrasts using this code block. And then to sort of probably make it a little more clear what I'm talking about, we can go ahead and view the contrasts table that we've just created. So you can see here we have a matrix with two columns. Each column header designates a different contrast that we're going to be performing. So go ahead and use this contrast table to answer the following questions. So how many comparisons are shown? So how many comparisons are we going to be feeding into DC2, and what are they? So go ahead and put your answers in the chat. Yes, exactly, Alan, that's right. So the code that we ran in this code block basically identifies all possible pairwise comparisons. Since we have two groups, there are two possible pairwise comparisons, flight versus ground control and ground control versus flight. So, exactly right. We're going to come back to question number 2 in a little bit after I've lectured on log-fold change. So the next thing that we are going to do is to import that RSEM raw gene counts data table that we just generated. And we're going to use, when we do this, we're going to use the a whole nested set of R functions to read it in. So First, we're using that file.path function to stitch together our counts directory and the name of the file that we're reading in. We're using the read.csv function again. And then, interestingly, remember read.csv generates a data frame data structure. So data frame is a very common type of data structure in R, but there's a kind of a simpler and cleaner data structure called a matrix. And so we need to convert the data frame generated by read.csv into a matrix using a function called as.matrix. So we're going to do all that and assign it to a variable called raw counts. And then because DC2 can only take a count matrix that has integers in it, we're going to use the function ceiling that we learned about to convert the raw gene counts table to only contain integers by rounding everything up to the nearest integer. So let's go ahead and run that. And then that cell, and then let's add another cell here and actually just view the first few lines of our raw counts table so we can make sure that we've done what we think we've done. So the code for that, of course, is just head, open parentheses, raw counts, close parentheses. And you can see we've read in what looks exactly like the data frame that we wrote out at the end of the FASTQ accounts notebook, except we've coerced everything to integers. So no decimal places. All right. So now we have our metadata, we have our counts data, but DC2 can't just take in data in those formats. Instead, DC2 requires a specific input called a DC2 Dataset Object. This is just a special data format designed for the DC2 package, and it has different slots for data. One slot holds the gene expression data, one slot holds the sample group assignments, one slot holds the different contrasts. And so we're going to use a function that's provided by the DESeq2 package called DESeq dataset from matrix. So very obviously what this function does, takes a matrix and creates a DESeq dataset object from it. And then that's going to convert our raw counts data table into a DESeq dataset object. So this function takes in a few different things. First, it takes in a matrix of counts data, and that's the raw counts variable that we just defined. Secondly, it takes in a metadata table describing which group each sample belongs to, and we read that in earlier and defined it as a sample table variable. And then third, the condition in the metadata table that DC2 is going to use to group the samples for comparison. So here we only have one condition column, and we use that to group samples as either flight or ground control for DG analysis. Right, so, and the resulting DESeq dataset object, which we're defining down here as our variable DDS, is going to hold the counts data and the group assignments for each sample in what they call a design formula. And so as we continue doing analysis with DESeq2, the differential expression results that we create are going to be added into the DESeq dataset object at different slots. So like I said, there are slots for gene expression data, sample group assignments, and then there are empty slots that are empty right now that we're going to fill in with differential expression data. So let's go ahead and run the DESeq dataset from matrix function to which we've passed our raw counts variable, our sample table variable, and then the name of the column holding the condition, which is going to be used to make the groups. Oh. an interesting error. Hmm. This is the sort of error one would get if one had not read in the DC2. Ah, okay. Rashi, did you run the cell that had ceiling in it?
I got the same as Rashi.
Oh, I see. So
So
you,
we reset. So resetting the kernel has something to do with it. We reset the kernel.
yeah, um, exactly. That's probably what happened. So, we read in the libraries, and then we went down and changed. I went down and said, "Oh, select R" as the kernel. And so it forgot that I had read in the libraries, which means that I need to read in all the libraries again. So anyone who reset their kernel, please go up and reload all of the libraries at the top. Anyone who didn't reset their kernel does not need to do that. Okay, Henley, I get T and F for exclamation point head raw counts. So Henley, in this notebook, we're running an R kernel and we're running an R function head. So we don't need to put an exclamation point. So I think if you just run head raw counts without any exclamation point, you should be able to see the counts matrix. Does that make sense? Okay, good. Yeah, that's a good question, Felix. When we actually run the DC2 part of the differential expression analysis, it's such a large process that we're going to send it to the Slurm job scheduler. And so we're going to have to run a Unix command to run the Slurm job scheduler. So when we get there, I'll point it out. This is how we run Unix commands from an R notebook. If you wouldn't mind holding on to that for a moment. All right, so did anyone not get this pink message converting counts to integer mode when you ran the DESeq dataset from matrix cell? Okay, good. So this is just a message that DESeq gives us, letting us know that our counts table has been read in as integers. So that's good because we converted it to integers. Now let's go ahead and use this summary function that we learned about in the intro notebook. Remember I said the summary function is nice when you're working with a matrix because it'll do some mathematical summaries. But if you have a more complex object, like DESeq Dataset Object, for example, that has data in all these different slots, it'll give you summaries of those slots. So let's go ahead and run that cell, and what it's telling us is, okay, DESeq Dataset Object, length of 55,487, 0 metadata columns, because we haven't added any metadata yet. So based on the output here, how many genes do you think are stored in the DESeq dataset object? Go ahead and put your answer in the chat. So based on the output from looking at the summary of the DESeq dataset object, Christina, rather than what we were talking about before. But just looking at what we've read in here and what summary is telling us. Yeah, so we also used, in the previous notebook, we talked about how we can use shape to look at the shape of the, okay, well, counts is not defined. We can look at the shape of the raw counts matrix, and we commented on how mice have 55,000, over 55,000 genes, and we have 12 samples. So when we take this counts matrix and we then feed it into DC2, we would expect, okay, we have the same number of genes, 55,487, and that is exactly what we see. So here length means the number of rows in the matrix that we fed into DSeq2. Right. So the next thing is that, we saw when we run DSeq dataset from matrix, everything works fine as long as we've run sealing when we read in the raw counts data. What happens if we don't? What happens if we don't coerce these data to integers? So the next thing is a challenge. I want you guys to use the next few code blocks to run DSeq dataset from matrix without first converting the counts data to matrix. So the way you're going to do this is pull in the raw data again, like we did at the end of step 1b, but make sure to use a different variable name so that you don't overwrite your raw counts variable. And then don't run sealing, and then run DSeq dataset from matrix again using your new variable in place of raw counts, and go ahead and store it in a different variable name so you don't overwrite your DDS variable. And I'll let you spend a couple of minutes with that and then have you put your code in the chat. Okay. Go ahead and put whatever code you have in the chat along with any error messages that you get. Nice, Zinru. Did anybody get a different error than Zinru and Henley? All right, great. So as we've seen, we can just read in the raw data again without running sealing. And then I've created a new variable RC, and we can run DSeq dataset from matrix on our new variable. And we get an error. So the background is pink both when we just get informational messages from DSeq2 as well as when we get errors, but it's very clear when it's an error because it tells us. And obviously the error is just, well, you've passed me some data that isn't in integer form, and it says clearly in the documentation that the data has to be in integer form. So very good. Felix says, if you wanted to see all the other things in DDS, you could run strDDS. That's right. You told us about this function earlier on. Let's see what this shows us here. Nice. Formal class, DC dataset from the DC2 package with 8 slots. Design, row ranges, number of rows. Here's our 55,000 number. The names of the genes are here. These are our sample names. Very good, thank you, Felix. I could imagine multiple scenarios where you want to pull out some of these, some of this information and look at it to troubleshoot or just to subset your data or something like that. Yeah. Thank you very much, Felix. All right, so let's do a little bit more data processing here. We've read in our counts data, it's over 55,000 genes, but remember a lot of these genes probably have really low expression just because of some of these genes were not being expressed at the time or not expressed in this tissue. And so those are really uninformative, especially for differential gene expression analysis. They're just noise and they extend the time that it takes to run these algorithms. So we're going to filter out genes that have a count sum of less than 10 across all samples. And remember, we can do this by using the row sums function in R. We can define a variable called keep, where we say keep is defined as row sums, and we're going to access the counts slot of the DDS object. We're going to say calculate row sums on every row in the counts object of DDS2, and let's keep only everything that has a row sum greater than 10. Then we're going to subset our original DDS object with the brackets, remember, which slice data, and we're going to add the comma where we are telling it we want to slice on rows, not on columns. So we can slice out just the rows we want to keep. Oh. Okay, can you scroll up to the last set? Okay, good. No problem. All right, let's filter out uninformative genes by running this cell. And then we can run summary again just to get a quick look at how the DC dataset object has changed. So based on the output here, how many genes had a count sum of less than 10 across all samples? Yeah, a lot. So, no worries. Yeah, so 33,625 genes had a count of under 10 across all samples, which is incredible to me. That's over half of the genes that we started with, but that speaks to the importance of transcriptional regulation, because we have, the same DNA in all of our cells, but not all the genes are turned on all the time, and we're just looking at the soleus muscle. So we have genes that are special and important to the soleus muscle, and A lot of them are lowly expressed and are not going to be informative for this analysis. And so now, kind of a freebie, how many genes are stored in our DC dataset object at this point after we've done the filtering? Yeah, exactly. 21,862. Still a lot, still plenty of genes to look at. Yeah, but now we know they're actually going to be informative. All right, so wait, I'm so sorry. Where did the good question, Eowyn. So the 33,000 number is how many genes did we filter out? So how many genes ended up having a count sum of less than 10 across all samples? So that number is the original, yeah, okay, the original number of genes minus the current number of genes. So just how many we filter. Yeah, no worries. Okay. Now, what I'd like you to do is use the next couple of code blocks to see what if we filter out genes with a count sum less than 20 across all samples or less than 50. And so you can go ahead and run the same code as in the last previous two code blocks, but make sure you use different variables. Don't overwrite your keep or your DDS variables. So I'll give you just a second, and then you can go ahead and tell me how many genes do you have left when you filter genes with a count sum less than 20 out or less than 50? And if anyone gets an error, please feel free to share that as well. All right, let's walk through how to edit this code a little bit. All right, looks like some people have, do you guys want to share your code in the chat? All right, awesome. So keep in mind that when you are rewriting this code, you have to make sure to replace every instance of the variable keep and every instance of the variable DDS. So I suspect that might be where your error is coming from, Eowyn. Awesome. All right, so I just renamed all my variables with a 20 for the filtering out everything greater than 20. So the object is 20 not found. Oh, sorry. That's right. The very first time you call DDS, you're calling the original DDS, and you're subsetting the original DDS. So I'm sorry, I misspoke earlier. You're not replacing every time you call DDS, because you're calculating row sums on the original DC dataset object, and then you're subsetting the original DC dataset object, but you don't want to overwrite it, so you write it to a new DDS variable. and I get the same number that you guys are getting for row sums greater than 20. What about row sums greater than 50? So what was interesting to me here was when we did our first pass at filtering, we went from 55,000 down to 21,000. That's a huge jump. But the second couple of passes, we haven't actually gotten rid of that many genes, right? We've gone from 21,000 to 20,000, and then down to 17,000, which is a little bit of a jump. But still, you know, only 3,000 genes. So I think what that's telling us is that, you know, we picked filtering on count sums of less than 10, not arbitrarily, but everyone has a different cutoff that they're comfortable with here. And so I think when you're not sure which cutoff to use for filtering, you can do this kind of stepwise filtering and see where is the point where filtering stops being all that informative. Where does the cliff fall off and you stop actually removing that many genes? So looking at this analysis here, I would say I think removing genes with fewer than 10 row sums across all samples is a pretty good cutoff because you don't end up gaining that much when you keep increasing. How would not filtering change your final result? A cutoff of less than 10 is suggested as best practices for the DC2 authors. Yeah, perfect. Yeah. So if we didn't filter, Felix, it's likely that the end result of the differential gene expression analysis would not change that much because the genes that are selected by differential gene expression analysis have to change pretty significantly between groups, which means that in some samples they have to be expressed pretty highly. However, there could be a couple of genes which are being filtered out that would somehow become differentially expressed because all of their row sums were over in one flight or ground control group. But we probably don't want those results because at this point, the values in our gene expression data, since they haven't been normalized yet, they range, as we can see here, from 9, 11, all the way up to 90,000 or 108,000, which means that the range of expression here is gigantic, and any expression that is low enough to get, any gene that is expressed lowly enough to get less than 10 across all samples is probably just noise. And anything that's as low as like 1 or 2 or something is probably just noise, and it's not something that we want to have in a high quality analysis. Does that answer your question? If we have time at the end, we should try running this Jupyter notebook all the way through without filtering. Sure. Mine's giving 17,653 for 10, 20, and 50. Christina, I wonder if you overwrote one of your variables. Would you mind sharing the code that you're using? Or I can put my code in the chat and you can see if that fixes your problem. The important thing is that you don't want to overwrite your original DDS object. So you might want to go up and recreate your DDS object. Yeah, Christina, see what happens if you recreate your original DSeq dataset object all the way up here, the DSeq dataset from matrix cell. And then I'll put my code in the chat. And then hopefully you'll get the same output. That fixed it? Oh, good. So you're getting the same output now, the different lengths now. Awesome. Amanda, does DC2 suggest a cutoff of row sums 10 no matter how many samples?
Yeah, so it doesn't specify a sample number. This is something that people have brought up, though, is that maybe we should make it a proportional to the number of samples, to make sure that every time we run a data set, it's respective to the number of samples of our cutoff. But pretty much you're just trying to move anything low expressing. So as you guys noticed, when you just increased it to 20 a little bit more, you didn't really change too much or get rid of too many genes. So that's why we just keep it at 10. But yeah, so their suggested practices actually doesn't really make a reference to how many samples you have, believe it or not.
Yeah, that threw me for a loop a little bit when I first started using DESeq2. I know other groups use, for example, instead of filtering on just a value, they will filter out the lowest expressed 10% of genes or 20% of genes. So that takes into account how many samples you have and the kind of the stochasticity within your data set. But as Amanda says, it seems to empirically be a pretty good cutoff. And yeah, good point, Amanda. Let's everybody create a new cell here, please, and run summary DDS. And let's everybody make sure that we are getting, so we are still getting 21,862 when we run summary DDS. Make sure nobody has overwritten their DDS object. All right, thumbs up. I like that. All right. Perfect. Okay, last step here is something that you will not have to do probably if you are running DESeq2 in general, but since the DESeq2 analysis is so computationally intensive that we're going to have to run it in a Slurm script, we need to actually write out the DESeq dataset object to a file so that we can so that it can be accessed by Slurm. So we're going to just use write.csv for that. We're going to convert the counts slot of the DDS object to a data frame by using as.data.frame, which is like that as.matrix function we used before. And we're providing a file path by stitching together the norm output file path variable we generated before and our new file name, which is just dds.csv. And we want to specify, yes, we do have row names in our data. So let's run that cell. All right, so that is as far as we're going to go in the Jupyter Notebook right now. I'm going to go ahead and switch over to the first part of the statistics lecture. to give us some of the statistical background that we're going to need for running and understanding the outputs of DC2.
I had a question about why we're filtering the stuff, because wouldn't we want the smaller counts essentially? Like, why would we want to filter out any of the code with the smaller or lower than 20 or lower than 50, or in this case, lower than 10?
That's a really good question. And the reason is we don't want to filter out anything lower than 20 or lower than 50. But the assumption is that if the gene expression is so low that it sums across all your samples and it still ends up being less than 10, which in our case would mean that basically probably there are 10 samples with a gene count of 1 and 2 samples with a gene count of 0, for example, that would, or 9 samples with gene count of 1 and 3 samples with a gene count of 0, that would get us less than 10. That, to a bioinformatician's eyes, is just noise. So there are no, if you get a gene that is expressed with 1 count, we don't care about that. That gene isn't doing anything. It's not really biologically active. It could be a technical error. In fact, it probably is a technical error. It's not changing the cell appreciably, because if it's real, there was 1 count of that gene, then it's going to be translated into 1 protein. So on a biological level, it's noise, and it's probably technical, and it will slow down the analysis considerably, given that it was over half of our data set was expressed that lowly. And with just one other biological point, it can make sense that genes are just plain not expressed in certain tissues, because they're tissue-specific genes, or they're not turned on at this particular time in the animal's development. That maybe they're embryonic genes or early development genes. So we're not getting rid of valuable information. Instead, we're filtering to preserve only the valuable information. Does that answer your question?
Yeah, that does. Thank you so much.
No problem. All right, so let's get into some statistics here. All right, so the topics that we're going to cover today and tomorrow, we're going to start out with some really basic sort of metrics that are used to quantify and describe data. These are, mean, variance, standard deviation, things that perhaps learned in high school or college math. But we're going to go over them because they form the basis for more complex calculations that we're going to get into later on in the lecture. We're going to calculate some of these by hand, but in bioinformatics, luckily for us, these are calculated by computational programs. So we're not going to have to actually calculate them by hand, but it's good to know the mathematical basis. Then we're going to move into some more sort of commonly used and a little bit more complex concepts and calculations in probability and significance. And you may or may not have run into these in a structured way unless you've taken a statistics course. And these are really important for understanding how we decide which genes we agree on are differentially expressed. And then finally, we're going to move into the 3 key methods that are used in the DC2 package. And Amanda has already touched on some of these. Mediative ratios method for normalization, maximum likelihood estimation, negative binomial model. These are much more advanced topics in probability and statistics, and it's fine if we take a little bit more time to go over them, if there are lots of questions. And these are things that even there are some biologists and bioinformaticians who still have to go look these up every time. So I really want to give you guys a good foundation so that you can understand the analysis that we're doing. So first of all, I wanted to just start off really simply by defining some important terminology, because if we all agree on the terminology, then we'll know what we're talking about. And all of these terms are about to be illustrated concretely with examples. But just to get some text on the page, when I say sample data or data set, this means a set of data points from an experiment. So in our case, we're talking about data from the soleus muscle. Our data points are samples that were taken from mice. A sample is a self-contained measurement of one or more variables. In our case, the variables are genes because they're gene expression data. A data point is a single value in a data set. For example, the expression of one gene in one sample. And then a variable is a quantifiable aspect of each sample. So in our case, variables are genes, but they can be numeric or non-numeric. So what I'm going to do for the first part of the lecture is, because gene expression data is large and a little bit abstract, I'm going to take a step back and talk about a different data set. And because statistics can be a little bit dry, I'm going to try to have a little bit of fun with this. So since we're going to be working with spaceflight data, let's just imagine that an advanced carbon-based life form named Mel sets out from a planet in the Tadpole Galaxy, which is a real galaxy, 420 million light years from Earth on an exploratory voyage. And upon arriving in Earth, Mel accidentally lands the spacecraft in a large field in Portland, Oregon, which is apparently known as the iris capital of the world. They have fields and fields of beautiful irises. And because all there is to see are irises, Mel thinks, well, this must be the dominant life form on planet Earth. And since this is an exploratory voyage, yes, Felix, that's what Mel looks like. Very good. How did you know? Spends a year collecting detailed data, petal length and width, sepal length and width, and Mel ultimately, after all the cataloging, decides that there are three main iris species and names them Setosa, versicolor, and virginica. And Mel then goes ahead and sends this iris data set back to the home planet and says, okay, I've characterized the dominant life form on planet Earth, and here are all the physical characteristics and the different species. And the scientists back home have a lot of questions. So here, what I'm showing are the first 5 samples of Mel's iris data set. And the reason that we're using this data set is because it is publicly available in R, and I believe that someone earlier mentioned that we could use this data set. So some people are already familiar with Mel's data set, which is great. So I want to just point out, for those of us who are not iris scientists, if we're talking about the petal, that's this part of the flower, so petal width and length, and then the sepal is this longer part, which supports and protects the petals, and then the petals surround the reproductive organs. So anyway, Mel develops this data set, and if we want to just stop and think about the important statistical data set terminology from earlier, okay, really good question from Naval. Is it more common to use features as rows and samples as columns? Yeah, in machine learning, we use samples as rows and features as columns. Yes, it is more common in bioinformatics to do features as rows and samples as columns. And so if you are transitioning a data set from bioinformatics to machine learning, you usually have to transpose it, because machine learning expects features as columns most of the time, probably because features are the most important thing in machine learning, whereas in bioinformatics, I think we think of samples as being the most important thing, and we're just using the features to learn about the samples. So great question. Yeah. Okay, so here in our data set, as Naval pointed out, our samples are columns, and then our features, which I also call variables, are the rows here. I think maybe variable is a bioinformatics term for features, whereas in machine learning it's features. All right, so samples as columns, variables as rows. The whole thing is called sample data or data set. And then there's one data point. And so as you can, we've been working with this gene expression matrix. And so you can imagine how this would map really nicely onto our gene expression matrix, where we have samples as columns and then each gene is a variable. Anyway, so Mel's friends back home want some basic descriptors of the different species on the new planet Earth. And so Mel invents a new quantification, the mean or the average or the mean of a set of numbers. So I know we all know how to calculate the average of a set of numbers, but just to have some text on the page, we add all the data points together, and then we divide the sum by the total number of data points. And this is a mathematical equation of the mean, where M stands for mean, X is a data point, and N is the total number of data points. So if we wanted to calculate the mean sepal length, obviously, for samples 1 through 5, this is how we would do it by hand, but there is also a function to do this in R, helpfully called mean. And we pass to this function a list of numbers. And remember, in R, the way that we create a list is using the C function, which stands for concatenate. So we just concatenate together these 5 values and pass it to the mean function, and R will calculate the mean for us. Now, Mel realizes that the mean quantification has some limitations. It only describes the center of a data set, but not the outer edges. And the edge cases are usually the most interesting. So Mel goes ahead and invents variance or dispersion. So one way of thinking about variance is that it tells you the consistency of the points in a data set. So that is, a data set with low variance has data points closer together or more consistent than a data set with high variance. This is important because if we know the variance, this will help us predict the likelihood of unusual events. How many unusual events are there? We predict how likely there is to be another one. Now, this is really important to use alongside mean and variance to describe a data set, because data sets can have the same mean but different variance. So for example, this mean is so misleading. Exactly. And here's a great example of that. We have a data set here where the mean is at 100. On a second data set, the mean is still at 100, but the variance is so much greater. So the left-hand side, the data set has values from 75 to 125, but on the right-hand side, the spread is much, much larger, even though the mean is exactly the same. So we'll use variance as a building block in the next quantification. But to calculate variance, the important thing is how we calculate variance depends on whether we're calculating variance for the whole population, which is usually impossible because it's like all irises on Earth, or all humans on Earth or something like that. We can't usually get all of those data points, or just a sample from the population, which is usually what we're working with. For example, Mel's irises data set, all of the irises that Mel was able to measure in a year. So really, the tadpoleish scientists want to know the variance for the entire iris population. just the same way, we want to know things about the whole human population. But the reality is we don't have time for that, and there are only so many researchers. So in this case, Mel goes ahead and samples a reasonable amount. And we hope that the sample is big enough to be representative of the population. And there is a part of statistics that deals with how big a sample needs to be in order to be representative of the population, but that's a little bit beyond the scope of this lecture, so we're not going to be going into that in this boot camp. It's important to think about whether or not your sample is big enough to be representative of the population, and to just be conscious that some samples are not large enough to be representative of the population, especially in biology, where sample sizes are necessarily small because it's hard to take samples and it's expensive. In any case, the technical mathematical way to calculate variance, we're going to focus on calculating the sample variance, which is again the variance of a data set sampled from a larger population. We start out by calculating the mean, using the mean as a building block here. We then subtract the mean from each data point, square each result, sum up the squared results, and then divide by one fewer than the total number of data points. So really what we're getting at here is, what is the relationship between the mean and all the data points? So in the two distributions we looked at earlier, in one distribution, the relationship between the mean and all the data points was, well, most of the data points are really close to the mean. And so when you calculate your variance is small. But in the other data set, there was a substantial number of data points that were far away from the mean. So in calculating the variance, you get that relationship between the mean and the data points, and the variance ends up being larger. So the equation here is again, V stands for variance. We sum up with a Greek letter, capital sigma for sum. We sum up all of the data points minus the mean squared over one fewer than the total number of data points. If we were interested in finding the variance of the sepal length for iris samples 1 through 5, again, the sample variance here is the variance of just these 5 irises, whereas the population variance would be the variance of all the irises. So if we wanted to calculate the variance of just sepal length for just these iris samples, by hand we would first calculate the mean, which we did already, subtract the mean from each data point, square each of those results, sum up those results, and then divide by one fewer than the total number of data points, which here is 4, in order to get a sample variance of 0.046. Fortunately, we never ever have to do this by hand, because R has a handy function called var for variance, to which again we pass a list of numbers. Interestingly, what you'll notice here is that when I calculated the variance by hand, it was slightly off from what we get programmatically, 0.046 rather than 0.043. Why do you guys think that is? Can put it in the chat. Any ideas? Yeah, exactly, Rashi. It's rounding, because I'm just one person and I can't be responsible for saving digits out to, the 20th or 30th place. So I rounded everything. But R has the luxury of being a computational program and doesn't have to do that. So that's another good reason to do all these calculations programmatically rather than by hand. All right, so at that point, the thing is, standalone variance kind of has limited utility on its own. So, 0.043, what do we do with that? So Mel has the same realization and realizes that as a standalone number, variance is a little difficult to interpret and visualize. So Mel realizes if you take the square root of the variance, the resulting value very nicely describes the average distance that each data point lies away from the mean. Really nice descriptor of how spread out the data set is. And so Mel calls this new metric the standard deviation, or SD. Very simple equation, square root of the variance. And the standard deviation is a measure of how spread out your data points are. And I'll point out here, we call standard deviation SD, or we refer to it as the Greek letter sigma. So here's an example of just a really nice, beautiful fake data distribution where we can see what is a standard deviation and how does it relate to the mean. So here the mean is delineated with the Greek letter mu here in the middle, and then we can see mu plus or minus sigma, or the mean plus or minus 1 standard deviation, 2 standard deviations, or 3 standard deviations. And this graph helpfully tells us that 68% of this particular data set falls within 1 standard deviation away from the mean. 95% falls within 2 standard deviations, and nearly 100% are within 3 standard deviations of the mean. So if you're able to describe your data set like this, it really gives you a nice understanding of, are your data all clustered around the mean, or are they more spread out? And on the level of an individual data point, the less, if you know where your data point is, you can say, well, this data point is so many standard deviations away from the mean. And that gives you an idea of how typical your data point is, because the less typical or the more unusual a data point is, the more standard deviations it will be away from the mean. So if we were interested in finding the standard deviation of the sepal length for our iris samples 1 through 5, we would simply take the square root of the variance we just calculated, or we would use the SD function for standard deviation in R. Again, very nicely, we just have to pass a list of numbers to the SD function. Okay, now we get into something a little bit more interesting. We so far have been looking at just the first 5 samples, right? Because I didn't want to do by hand calculations of anything more than 5 samples. But it's a lot more interesting to look at the distribution of, let's say, all the sepal length data. So what we're looking at here is a histogram of sepal length for all iris samples. I believe there are 150. We know that the mean sepal length across the whole data set is 5.84, and the standard deviation is 0.82. I've colored the distribution by the different species. So Setosa is pink, first color is green, and Virginica is blue. You can imagine that in another situation, for example, flight and ground control would be analogous to species here, and you could color your histogram by those groups. And the lines here show where the mean is, and then where plus or minus 1 standard deviation away from the mean, or plus or minus 2 standard deviations away from the mean. The x-axis shows sepal length in centimeters, and then the y-axis shows number of samples. So to start to sort of interrogate this data set and understand what these different statistical terms mean for us as researchers, I want you guys to help me answer a couple of questions using this plot. So first of all, is the standard deviation of all the data greater or smaller than the standard deviation of just the first 5 samples? And why do you think this is? So we can recall that the standard deviation of just the first 5 samples was around 0.2. whereas here, it's kind of laid out for you here. Go ahead and put your answer in the chat. Yeah, exactly. Why do you think the standard deviation of all the data is greater than the standard deviation of just the first 5 samples? What does standard deviation measure? Yeah, exactly. Broader range, it's the spread of the data, more variation. Exactly. More samples, more variation. A subset, especially I took a subset which was all from one species, so it's all going to be pretty similar, whereas here we have 3 species. Yeah, exactly. The full data set has more spread out data points. Yeah, more opportunity to include an outlier, exactly. Now, let's zero in on some of the interesting patterns just within this data set. So, for example, let's take a look at the data in between the mean and 1 standard deviation less than the mean. Is there a species-specific pattern? And what about, similarly, the data between the mean and the mean minus 2 standard deviations? What do you guys see there? Is there one species that's more represented? Nice. So mostly Setosa. Exactly. Yeah, and Alan notes we find more versicolor in between the mean and mean minus 1 standard deviation than we do between mean minus 1 and mean minus 2 standard deviations. Yeah. But in general, the Setosa species seems to be kind of left of the mean. So it's making up the majority of the samples that we find left of the mean, especially if we look at the mean minus 2 standard deviations. So let's do a thought experiment then. What would happen if we got rid of all the Setosa samples? What would happen to the mean of the data set? Would it increase or would it decrease? What do you guys think? It would increase, exactly. And that's because we would have gotten rid of all of these lower values. So then, can we use this graph to answer the last question? So based on this graph, are we seeing that one of the iris species appears more different than the other two? It's more separated out than the other two. And if so, which one? Okay, I see a couple of Virginicas. Does anyone have a different interpretation? And we can talk about it. So this is very visual and a little bit subjective. And so we're going to get into, yes, can we further quantify this? Eowyn, jumping ahead. The reason I'm leading us down this trail is to get us to ask exactly that question. This is a little bit hard to analyze visually. Appearance-wise, it's hard to say. When I look at this distribution, I personally say, I think Setosa is a little different. I think it looks like Versicolor and Virginica are overlapping with each other more than Setosa is with either of them. But some of you have interpreted it differently. So can we come up with what if we wanted to quantify the differences between these species? So to do kind of a pairwise comparison between all these species, figure out, is one of them different from the others, or are they all equally different from each other? So Mel ends up coming up with exactly the same question and discovers that you can use the fold change metric to quantitate a difference between two groups of samples. Fold change is really simple. To calculate the fold change between two data points, for example, x and y, you simply take the ratio of x over y. So for example, if we wanted to calculate the fold change between the data points 6 and 10, we would take 6 over 10, and you would get 0.6, and we would say there's a fold change of 0.6 between 6 and 10. Now, we're going to get into a little bit of a more concrete way of using this and a better way to interpret it, but I just want to say the reason that this is interesting is because if these two values were exactly the same, we would expect the value of the ratio to come up equal to 1, right? So if there's no fold change at all, if there's no change at all, your fold change value is 1, so that's no change. And the more different the two values are, the smaller the fold change gets as it moves away from 1 closer to 0. So that's just kind of baselining what we expect from fold change. Why did you choose division instead of subtraction? That is a good question. Let me just think about that for a second. I think it's because of what I just said about ratios being easy to interpret. So if you do, if you subtract, then you can get whatever number you want. Yeah. Phil is raising, Dr. Heller is raising his hand to help me through this. Please go ahead.
Fine. It's fine to call me Phil. The reason we don't do subtraction is just imagine if you've got 2 of something and the other data point is 4, you're doubling. That really means something. But if you've got 1 billion of something and 1 billion of 2 of something, that's hardly any difference at all. And the fold change by dividing will show that those two are very different situations, that from 1 billion to 1 billion and 2 is almost no change at all. But if you're subtracting, there's no way to tell the difference. Both 2 to 4 and 1 billion to 1 billion and 2 would have the same fold change if you define fold change by subtraction.
Beautifully said. Thank you very much. What if x is greater than y? We're going to get into that in a couple of slides, if you wouldn't mind holding on to that question. Thank you. So this is very, very simple. What if you have only 2 data points? Generally, we don't have 2 data points, though. So in general, what we want to know is the fold change between 2 groups of samples. For example, we were just talking about the iris species. We want to quantitate the difference in sepal length between groups of iris samples. And so in that case, what we do is to calculate the mean value of whatever the variable of interest is, the sepal length or some gene for each group, and then we take the fold change of the mean values. So for example, we could calculate the fold change of sepal length between all of the pairwise iris species comparisons. Here I'm showing what it looks like if you view the entire iris data set in an R Jupyter notebook. You'll notice that there's some ... here because Jupyter didn't want to print out all of the data. So if we want to calculate fold change just of the sepal length between all the iris species, first we want to calculate the means. So the first thing that we need to do is I used some RMagic. I nested some functions here, which basically gets a list from a row in a data frame. Because if you guys remember, if you just subset out a row in a data frame using the brackets for slicing, you just get another data frame. We needed the actual values. Remember, that's why we had to use the $sign. But here I specifically needed to get a list from this row, and then I pass that to the mean function using, I just subset to Setosa species, and I'm using only the sepal length values. So the mean sepal length for all the Setosa samples is 5.006. For all the versicolor samples is 5.9, and for all the Virginica samples is 6.588. Okay, so we have the mean sepal lengths. If we were interested in calculating the fold change of the sepal length between all the iris species, we would then simply, and I've done this again in an R Jupyter notebook, I've taken the ratio of the means, rounded to 2 digits using the round function in R, to which you pass the value you want to round and the number of digits to which you wish to round. And first I've done Setosa over versicolor, then Setosa over Virginica, and then versicolor over Virginica. So this is getting a little bit easier to interpret. If we were to look at these fold change values, based on what we talked about before, if your ratio is, if your values are exactly the same, your ratio is going to be 1, the closer you get to 0, the more different your values are. Which iris species are the most similar and which are the most different based on these fold change values? What do you guys think? Which two iris species are the most similar in terms of their sepal length? Yeah, exactly. Most similar, versicolor and Virginica, because their fold change is 0.9, it's almost 1. What about which 2 species are the most different based on their sepal length fold change? Setosa and Virginica, right, exactly. Exactly. So we've now done the quantification that Eowyn was asking for when we were looking at the distribution before. We've noted that if we use the fold change metric, Setosa and Virginica are the most different, and versicolor and Virginica are the most similar, which, looking at the distribution, I think does make sense, but it was difficult to see just from looking. Is there a way to compare all 3 against each other? So there are ways to do that. That is one of the reasons why I did all the different pairwise comparisons here. So if we take all of these data together, we've described every single comparison that we could possibly want to do. So Setosa versus the other two, versicolor versus the other two, Virginica versus the other two, yeah. Now, fold change is nice, but imagine a situation where the values are so vastly different that the fold change becomes very, very small. Oh, is Dr. Heller's hand up?
Oh, sorry, I forgot to take it down.
No problem. It beeped at me the last time you raised your hand, and so it really got my attention. So I didn't notice it was still up. Yeah, thank you, Amanda, for noticing that. Okay, so fold change is nice, but as you can imagine, if we are talking about a quantification method that can go from 1 down to arbitrarily small, because it's never actually going to reach 0, but it can get very, very, very small depending on the difference between your two values, you can end up with a situation where the values for fold change are vastly different, especially in a situation where you're looking at a lot of different variables. For example, thousands of genes, which can have very different levels of expression, as we saw before, all the way from the tens to the hundreds of thousands. So your values for fold change, even though they're stuck between 0 and 1, can really be very, very different, which makes them difficult to compare and visualize directly. So in practice, we usually take the log of fold change values in data science. We call this log fold change, and it's usually abbreviated LFC. This is very common in gene expression data. What's the LFC of this gene between group A and group B? So for example, I'll go into why this is a little bit easier to interpret. For example, if we were to take the log fold change of the Setosa versicolor comparison, we would round our value to 2 digits, but we would take instead the log of the ratio of the means. Excuse me. We get negative 0.17. And then if we were to, I note here, in this case, when we take the log, a negative log fold change indicates lower overall values in the group being compared. And the group being compared is the group in the numerator. So here, Setosa. Then if we were to swap this comparison and do versicolor compared to Setosa, so this gets to Zindu's question of what if the value on top is greater. If we do versicolor versus Setosa, we get the same number, 0.17, but the sign has changed. So positive log fold change indicates higher overall values in the group being compared. So that's the numerator, and here that's versicolor. So log fold change is just a little more intuitive to interpret than fold change. So if we see a negative number in log fold change, we know that means lower. If we see a positive number, we know that means higher. And the opposite comparisons are just the negative inverse of each other rather than being just different numbers on the number line between 0 and 1. So before I move on to exploring some visualization methods, are there any questions about log fold change or anything else we've discussed so far? Is anybody lost? Okay, great. We're all log fold change iris experts now. Perfect. Okay. The thing is, what has Mel been ignoring so far? If we take a look at the iris dataset, Mel has been working with sepal length, right? So what has Mel been ignoring? Can you guys tell me? Petal data, all the other three measurements, sepal width and petals. Yeah. No, exactly. So Mel has been focused on one variable, or in machine learning terms, one feat, and has been ignoring the other three variables. So here I want to introduce the term multidimensional, because this iris data set is multidimensional. It has more than one variable. When I say multidimensional in this context, one dimensional means one variable. And that's important because gene expression data is also multidimensional, where each gene is a variable. Each gene is one dimension, and we'll come back to that later. But it's important to get into some methods to use all the variables together because that's the best way to use all the data you have. So how can Mel work with all these variables and more completely analyze the differences between the three iris species? Well, one thing that Mel could do would be to create a two-dimensional scatter plot. So this would show the relationship between two variables. So, for example, this 2D scatter plot has sepal length on the x-axis and sepal width on the y-axis, and each of the dots here is one of the iris samples, and they're colored by different species. And this is great, but it's only two variables, right? It is possible with some real fancy coding to do a 3D scatter plot and visualize the relationship between three variables. Yeah, that's what I said when I first saw this plot. Anyway, here we have sepal length on the x-axis, petal length on the y-axis, and sepal width on the z-axis. This is a little hard to interpret the differences that we saw in the 2D scatter plot, where Setosa is kind of separated from Versica and Virginica, replicating what we saw in the histogram earlier. Those differences are really kind of being obscured here. I imagine if this were interactive and we were able to turn it around, we might be able to see a little bit better. Yes, Felix, you could do a movie for 4 variables. That's right. Can you do a movie for like 30,000? Or even like 10 or something like that? I'm actually asking. I don't know what technology is necessarily out there for real-time visualization. Depends on what we're trying to show with the comparison, correct? More variables on one graph isn't necessarily better. Right, right. That's true. Movies with multiple screens, good. Time can only take care of one dimension. That's true. So, in the Tadpole Galaxy, I think they didn't know that you could do a movie for 4 variables. So Mel gets really ambitious and invents principal component analysis, or which we generally refer to as PCA. So this is PCA basically is a way of visualizing your data by taking into account all of your variables, doesn't matter how many, on a static two-dimensional plot. So the way that PCA accomplishes this is to reduce the number of dimensions, again, variables, by condensing all of the variables into a small number of vectors. And so in this case, when I say vector, what I mean is a vector is a summary of important characteristic information that previously was spread out across lots and lots of variables, but can actually be condensed into one vector or a small number of vectors. And these vectors represent the main sources of variance across the data set. So you can imagine you have a data set with a lot of variables, but especially in, let's say, biological data, gene expression data, these variables are not necessarily independent. Some of them vary together. So there are sources of variance that are consistent across many different variables. So you can take those sources of variance and condense the information contained within those variables down into one vector, and then take different sources of variance and condense it to another vector, and so on and so forth. So you end up with a few different vectors that really just represent all the interesting stuff going on in the data set without having to act on the level of your maybe 30,000 variables. And each vector is called a principal component, or a PC. And high dimensionality data, such as gene expression data, are usually visualized with a scatter plot of the first two principal components, because the assumption is, which has been empirically demonstrated to be true, is that the first two principal components generally represent the majority of the variance across the entire data set. So you're showing most of the interesting information in the data set if you look at a scatter plot of the first two principal components. So here is a PCA plot of all four iris variables having been condensed down into two principal components. I apologize for the quality of this image. On the x-axis is principal component 1. The y-axis is principal component 2. One sample is represented by one dot in the scatter plot. Within parentheses here on the x and y axes labels, we have the percent of the data set variance captured by the first and second principal component. So this is one of the key aspects of principal component analysis. We know what percent of the variance of the data set is captured by each principal component. So when I was talking about taking the interesting information in the data set and putting it down into one vector, one principal component, we actually quantify what of all the variance in the data set is actually captured by that principal component. So in this case, principal component 1 is capturing almost 73% of the variance, and principal component 2 is capturing almost 23% of the variance. So if you add those together, that's almost 96%. So this PCA plot is capturing nearly all of the variance in the data set, almost 100%, and is therefore very useful to show how the data in these variables combines together to show structure within the data. And when I say structure, what I mean in this case is you can actually see separation in the data after we've combined together all the different measurements, the sepal and the petal measurements. And you can see a very strong separation between the Setosa species and the versicolor and virginica species. You can also see a little bit of structure within this part of the plot where versicolor and virginica are combined, but they're not exactly overlapping, right? The versicolor data points are off to the left a little bit, virginica's down to the right. So PCA plot is showing us similarities and differences between data points in our data set. And that's one reason it's very useful. Another way to say this is that PCA is really good at finding clusters of similar samples. So for example, here we could call the Setosa cluster cluster 1, and we could call the combined cluster cluster 2. In this case, I manually assigned these clusters because they're visually pretty obvious, but you don't have to do that, and you generally don't want to do that. There are bioinformatic tools which will derive clusters from PCA plots. Before I get into hypothesis testing, I really just want to dwell on this idea that PCA is a way of taking very large, multi-dimensional, multivariable data sets, identifying the interesting variants in the data set, condensing that down into principal component vectors, and then putting that information on a two-dimensional plot so that we can visually identify structure within the data. So I was going to say, are there any questions? And I see that there is already one. How do you calculate the PC vectors? I am not in a position right now to, off the top of my head, talk about this. I can come back tomorrow with a nice explanation. Nival says a lot of linear algebra. That's true. That's about as detailed as I could get about this. Nival, do you have anything else to add, or have you delved into how to calculate PCs? I have in the past looked into this, but again, you don't do this by hand. You do it programmatically, so it's not something I have at the top of my head. If you want to unmute and talk about it, please be my guest.
We didn't really like calculate or anything, but I learned about it in machine learning, and it was just like transforming vectors and stuff, and like we're doing orthogonal, like I forgot what it's called, orthogonal stuff. Yeah, so I just kind of learned about it a little bit. I didn't calculate it.
Okay.
Yeah.
No, that's cool. It sounds like you had a really good machine learning class. Everyone suffers through the manual derivation once, then uses R Python to do it after. Yeah, my manual derivation was like, 7 or 8 years ago, so that has whoosh gone out of my head at this point. Yeah, I will tomorrow come up with something that approximates a detailed explanation without getting us too much into the weeds, just so we kind of have a nice understanding of how the PCs are assigned and calculated. But that's a really good question, Zinru. Thank you.
So I took the same class as Nival had, and according to my notes, to calculate it first, you gotta find the direction in the data with the highest variance, and then you rotate the X and Y axis. Wait, no. First you gotta. Sorry, let me start from the top. First you find the highest variance in the data of the first feature, and then you find the highest variance of the next feature, I think, that's orthogonal to it.
Oh, yeah.
You kind of just repeat something like that.
Yes. This is all coming screaming back to me now.
The point is, it's linear algebra. That's it.
Yeah. No, thank you. Orthogonal, yeah. No, thank you so
Yeah,
much for
that
bringing
word.
up your notes. Thanks for bringing up your notes on that. That was really helpful. Felix says I'm more used to PCA, but do you use TISNE as well for visualization at GeneLab? I've heard it's popular in bioinformatics. So, Amanda, feel free to jump in. As far as I know, we don't currently use TISNE for visualization, but we will when we have a pipeline for single cell RNA sequencing analysis, which is something that the interns are going to be helping with this summer.
Yep, you got it exactly right. So TISNE plots are more commonly used to visualize single cell data or spatial transcriptomics data. So that is going to be part of the pipeline that some of our interns are going to be helping to develop.
Yes, exactly. Lucky interns. I know. Is your hand up, Dr. Heller?
Yeah, I am Phil and my hand is up. Can I just share a 1-minute intuitive description of PCA that I find really useful?
Please.
Can you go back? You've got a 3D plot of these points, one or two, back one more, back one more, back one more. There we go. So imagine that's not a plot. Imagine that I've actually got, you come over to my house and I've got this fish tank. And instead of fish in the tank, I've got these dots. And the tank is totally made of glass. You can walk around it. You can look at it from any point of view. What point of view is going to be most useful for you to understand the patterns in this data? And bear in mind, you can't perceive 3 dimensions. You've just got flat backs of your eyeballs. So you can only see a 2-dimensional picture. But what's the best 2D picture? What's the best angle of sight that's going to tell you what's going on? And the best angle of sight is what we get if you could now scroll down to the picture of the PCA. That's the best sight angle. There's one place you could stand in my house where you'd look at the fish tank and this is what you would see. And when you look at it this way, there are fewer points hiding behind other points. There's minimum shape and interesting stuff going on that you can't see. Most of the interesting stuff is visible here. So there are places where you could stand in my house where you wouldn't even see this separation between red, green, and blue. You could pick a bad side angle where they all seem kind of mixed together and very clumped together. This is a really informative sight line into my fish tank. So I think of PCA as the way of finding the best sight line on my fish tank, even if my fish tank is in 4 or 5 or 50,000 dimensions.
That is a really fantastic explanation, Phil. Thank you so much. It's just really, really intuitive. I can't really visualize a 50,000 dimension fish tank, but I guess that's the whole point. We can visualize a PCA plot of it. Thank you so much.
And by the way, that's another benefit of, 2 dimensions, as you said, is good enough for PCA. We don't really need 3. What that also means is that a 2-dimensional plot is a nice visualization. It's useful.
Yes. Yes, Amanda?
I was going to say, if you want to, feel free to hop to a web browser and open up GLDS 104 and click on that visualization link. You should be able to see the 3-dimensional PCA plot that you can play with and turn around to kind of put an image to what Phil is just describing.
Okay, you may have to guide me there. Visualization? Was that right? Amanda?
Yep.
Visualization? Okay.
And then you're already on PCA, and it takes just a minute to load. And then it should bring up an interactive PCA plot.
And it looks like Eugenie and Jonathan both shared the same video on PCA. Thank you so much.
So you should be able to go ahead and grab that and move it around.
This is spoiling the results of our PCA that we're going to do in the DG notebook later.
Probably
But it's so
a different
pretty.
data set.
No, this is a good one. Yeah, so what a great advertisement for the GeneLab visualization portal. We have 3-dimensional PCA plots. You can see there are certainly better places to view this plot from a 2-dimensional standpoint than others. Like this, you might think these 4 samples are actually all in a cluster together, but when you rotate it, you realize, whoa, they're not at all. There's 2 up here and 2 down here. This is fantastic. So this is kind of imagine you're walking around a fish tank. For sure. This is great. Can I put this link in the chat maybe? Let's see.
Yeah, for sure.
All right, if you guys want to play around with that 3D plot a little bit more, I just put the link in the chat. All right, and thanks for the clarification, Kevin. Appreciate it. Any other questions or thoughts on PCA? All right. A lot of votes in favor of StatQuest videos. Very good. Okay. I'm going to move on, but we can always come back to this. And we're going to do a lot of PCA plots tomorrow, so we'll have plenty of time to discuss. So, when you see a plot like this, the first question that comes to my mind as a bioinformatician is why? Why are these samples separating the way they are? And when you're looking at a PCA plot, we're talking about we've merged in hundreds or thousands of different variables. So it's not as if the two variables are on the X and Y axis. You would say, oh, well, it's because these, Setosa has much shorter leaves or whatever. And so I generally use PCA plots as a way to ask these questions. What is causing the separation? And so Mel, our imaginary friend from the Tadpole Galaxy, does the same thing. So having seen this PCA plot, Mel, who already knows about DNA somehow, takes a look at this plot and says, okay, I think that the differences observed between the iris species could be due to a gene that's turned on in the Setosa flowers but turned off in versicolor and virginica. What is the name for this type of statement in science? What do we call this kind of statement? Do you guys know? Yeah, exactly, Eowyn. This is a hypothesis. So this is my smooth segue into talking about hypothesis testing, because this is something that happens a lot in science, and this is exactly what we do in differential gene expression analysis. We're testing the hypothesis called, is this gene differentially expressed? But then we do it thousands and thousands of times. So before we get into that, we have to all understand what technically is a hypothesis. And the technical definition of a hypothesis is it's an educated guess. It's based on some data that you have, but you're not quite sure. It has to be testable in some way, otherwise it's useless. And for example, I want to remind you all of the sheep hypothesis from Phil's statistics lecture. So the hypothesis was all sheep are white, because maybe you've seen some white sheep, and so you think, okay, well, all sheep are white. Lots of white sheep support this hypothesis, but one purple sheep is enough to disprove it. So that's how hypotheses work. You can have lots and lots of data that support your hypothesis, but if you get some data to disprove it, that's actually more useful than data that support the hypothesis. So we're going to talk about in a minute whether or not it's ever possible to actually prove that a hypothesis is true. But first, let's talk about the accompanying statement that goes with your hypothesis, and that's called the null hypothesis, also sometimes referred to as H naught. So if we remember from a previous lecture, the null hypothesis is sort of the current or existing explanation, so whatever we're operating on now, or in science, the null hypothesis can just be coincidence. So the null hypothesis could be whatever we're observing is just coincidence. So for example, remember Daenerys decisively disproved the null hypothesis dragons are extinct by finding some dragons. But in science, it's almost never that decisive. So for example, if we go back to Mel's in science, since things are generally not so straightforward as they are in Game of Thrones, how do we go about testing hypotheses? In order to accept our hypothesis, it's not enough to just have a lot of supporting evidence. We have to actually reject the null hypothesis. If we usually can't decisively disprove the null hypothesis, because there could always be more data out there, how do we ever reject it? Well, the best practices are, first, we design and perform experiments to get data that supports our hypothesis. Then we perform statistical analysis on the data that we've collected to see whether our results occurred by chance. And if we can do enough to demonstrate that most likely a reasonable person would say that our results did not occur by chance, then we can go ahead and reject the null hypothesis. So for example, what would the null hypothesis be for Mel's hypothesis? So Mel's hypothesis, I reworded it here. I hypothesize the differences I observe between Setosa and versicolor and virginica are due to a gene that is turned on in Setosa but off in the other species. So one null hypothesis could be the differences observed between these species are not attributable to a gene turned on in Setosa but off in the other species. So it's basically just the opposite statement. So what's a good experiment that Mel could perform to test this hypothesis? Well, since we are in an RNA sequencing bootcamp, I would suggest that Mel could go ahead and extract RNA from several replicates of each iris species and perform RNA sequencing and search for a gene that's turned on in Setosa but off in the other species that could explain the differences that are being observed. Let me just see. Okay, 2 more slides and then we're going to end for today. But they're very important slides. So let's talk about, I said in the previous slide, one of the important steps for rejecting a null hypothesis is to perform statistical analysis on our data to see whether our results occurred by chance. Well, what does that statistical analysis look like? It looks like calculating a probability value or a p-value. And the p-value is the probability that your null hypothesis is true given all the data that you get from your experiment. So you want your p-value to be really small because you want the probability, I mean, if you're going to reject the null hypothesis, you want the probability of your null hypothesis being true to be very small. So in other words, the lower the p-value, the less likely the null hypothesis is to be true. In science, we generally accept that a p-value of less than 0.05 is low enough to go ahead and reject the null hypothesis. Because if you have a p-value of less than 0.05 and the p-value is the probability that your null hypothesis is true, then p-value of 0.05 means there's only 5% chance that your null hypothesis is true. So in that case, we say that chance is low enough that we can reject the null hypothesis and go ahead and accept an alternative hypothesis, like our hypothesis. And another way to put this is that a p-value of less than 0.05 indicates statistical significance. So that is, that the outcome of our experiment was likely not due to random chance. Now there's only a 5% chance that our experiment was due to random chance. So last slide here, very important question. Given everything we've just discussed, is it ever possible to definitively prove that a hypothesis is true using hypothesis testing? What do you guys think? Exactly. No. You cannot ever definitively prove a hypothesis. And as Eowyn says, you can only disprove a hypothesis. So the best we can do here really to get at disproving the null hypothesis is to assign a probability to the null hypothesis being true, and then say, well, we as a scientific community accept that at less than 0.05 probability, we're allowed to reject the null hypothesis. All right, so we're going to stop here. Pretty good place to stop. We've covered lots of the important stuff. We'll do a little bit more of the statistics lecture tomorrow and then do some of the Jupyter Notebook. In the last 2 minutes, does anyone have any burning questions about what we've just covered? All right, awesome. Thank you so much for sticking with me through statistics. If you think of some more questions, we can go over them tomorrow. Oh, you're welcome.
Hey guys, just before we sign off, I really want to stress that last point that Lauren just said. We cannot prove anything in science. So one of my biggest pet peeves is you will hear scientists tell you that they proved their hypothesis, and I cannot stand it when I hear scientists, especially well-respected scientists, say that they proved something in science. We can't do that. So it's a big pet peeve of mine, so I really wanted to nail that home. And also, I know that we owe you guys some stuff from your questions, so we'll make sure that tomorrow I have a calculation example of how RSEM calculates both gene length and expected gene length. Lauren will put together a simplified explanation of calculating principal component analysis, and I'll also explain to you guys a little bit more and show you some examples of what I meant when I said that some of those ensemble IDs, multiple ones can map or can indicate the same gene. So I'll make sure that I have some examples of what I meant by that tomorrow. So have a great night, and we will see you all back here at 9 a.m. tomorrow morning for our last day.